Hi, this is 3 Minute Bio with Luke D. Today's lesson is transcription.
All right, so we got 3 minutes to explain transcription. We're going to start with where this is all happening. So we've got a cell and it's got a nucleus. There's DNA inside that nucleus. And what we have to do is get information from that nucleus elsewhere. So parts of the cookbook are going to be copied and we're going to make recipes which can then be transported throughout the cell. A lot of those things are going to be taken, given to ribosomes, which are going to assemble a protein from them, from those instructions, but there are also other functions. RNA can act as an RNazyme, so as an enzyme in itself. It can also travel to other parts of the DNA and shut down or activate them. So let's see how this works. There's basically 3 steps. It's not terribly complicated. If you divide a gene into 3 parts, the beginning, the middle, and the end, essentially 3 things, or different things happen at each part. In the beginning, the first thing that's going to happen is some proteins called transcription factors are going to be activated by some process in the cell. Those transcription factors are going to travel to this beginning, or promoter. Now the transcription factor is specific to the promoter, so there's about 20,000 genes, and each of those genes, or many of those genes, are going to have different promoters. So if we were talking about OPRM1 here, OPRM1 would have its own transcription factor. That transcription factor, which is specific, is going to recruit an RNA polymerase, which is not specific. So RNA polymerase, an enzyme, is going to attach to the transcription factor and therefore the promoter. Now again, the transcription factor is specific to whatever this gene is. The RNA polymerase can be any RNA polymerase. The RNA polymerase is then going to travel forward. It is going to use small bits of high energy molecules, UTP, CTP, ATP, and GTP. It's going to use those to split open this molecule and make a copy of one of the strands. Now that's basically it from transcription. The RNA polymerase is going to travel to the end, which is the terminator, and then it's going to fall off. So really, the beginning and the end of the gene are kind of regulatory elements. The middle is where the code is that's going to be transferred to RNA. That's it.